A lot of these slides I've never presented before. I pulled them out of the archives about 2014. I had a phase where I was just writing down notes constantly, drawing slides, drawing equations, and I've never shown them in public before. They led to Lifespan, the book. They've led to pretty much all the work that we've been doing in my lab since. And I still use these thoughts to drive my lab's hypotheses. And it's a very different way of thinking than most labs when it comes to aging. And I'm happy to share with some of them with you today. And what I was actually trying to do was to come to grips with a lot of experiments that weren't making any sense at the time. That's often the way science goes, right? You have a paradigm shift that's necessary because the current paradigm just doesn't make any sense. And some of the things that weren't making any sense were that you could increase DNA damage in a mouse and it would live a normal lifespan. But in my lab, we were creating special types of DNA damage and we were accelerating aging. So how do you reconcile that? What is it about these Sirtuins that we've been working on since I was at MIT? And what is it? Is it really just that we break down, or is it something more than that? And through a wormhole, a deep dive, I would say, I got really deep into information theory. I got deep into the work of Claude Shannon and his colleagues, and even earlier. And things clicked. They started to really make sense to me that aging isn't so much a linear process, it's a circular process. And that aging is unlikely just to be wear and tear, but it's actually a software problem. And I began looking for ways to test that. And we still continue to test that. There are times in my lab where we still have confusing results, and we're challenging this hypothesis all the time. But compared to 10 years ago, we are much more, I should say, I'm much more enthusiastic about it because it really is, it's explaining a lot of things that have been going on, not just in my lab now, but globally. And you'll hear more about that from Vadim about epigenetic clocks. And we've been working in my lab on epigenetics since day one, and even before I had a lab. And so let me share some of that with you. This is going to be the first talk, I don't know, maybe ever, that I'm not going to show any data. I'm happy to tell you about what we're up to and where we're going and what the latest is. And I have some slides, but I decided this is a different group of people, different meeting. So let's talk about something that's a little bit more high level than messing around with cells and mice. You'll probably get a better insight into my brain than ever before. So this is an old yeast cell. I took a picture of it when I was in my 20s. I was working with Lenny Guarente, who I owe a lot to, my professor at MIT. And this is an old yeast cell. And what Lenny and I co-published in Cell in 1997 was that two major things happen for yeast aging. One was it seems to be somewhat of a program in that there's a stochastic element where you get genomic instability, the DNA breaks, you get a circle formed of ribosomal DNA. But after that event happens, now it's a very predictable program. We could mathematically model and predict the lifespan of yeast cells and their offspring. And all that math is actually buried in the back of that paper. And what we proposed was that the genomic instability in this organism led to the distraction of gene regulatory factors, and it was messing up the epigenome. And that gave rise to the phenotypes of aging, not just the death, but the phenotypes or the characteristics of aging in yeast are similar to us. They get old, they get slow, they get large, as you can see with this yellow acidic cell stained with bromophenol blue. They become sterile, and we could change the lifespan of a yeast cell by creating these circles of DNA or breaking the DNA in just a few defined places that would accelerate aging, including the sterility and the size and the slowness. And we could do the opposite. We could actually slow down the formation of these. And other colleagues in the lab made mutations that prevented circles from forming as frequently. And as the model predicted, the yeast cells lived longer and maintained their fertility. And so that led to a real shift, because at the time in the 1990s, when I arrived at MIT, the idea was that aging was caused by damage. So we expected in genetic screens to find antioxidant enzymes, but instead we found genes that controlled other genes, silencing proteins, which we now call sirtuins. There's a lot known about sirtuins, and there's not a lot of people, but there's a few people who still claim that it's debatable. It's not really debatable. We know how these enzymes work. We know how to activate them with small molecules, as you can see in this review by Michael Bunkowski and I. And so, I've spent a lot of my career trying to help people be healthier. So I've been co-developing molecules that can activate these enzymes. So you don't just have to fast or exercise to get the benefits. So these enzymes, what they do in yeast and in humans, in large part, is they stabilize the epigenome and they repair broken chromosomes. And they actually physically move, we think, between silencing and controlling genes and repairing DNA. And that's important for the hypothesis too. But what's important about sirtuins, named after the original Sirtu gene, is that Sir stands for information. And so that's obviously been driving my quest to understand what is it about information that's relevant to aging. So this is a series of slides that I drew in the early teens of this century. And so, these are ideas. These are not published. You can see what kind of things that I was thinking about at the time. And I was thinking, maybe we just need to revise the old paradigm. And instead of thinking about stages of damage leading to aging, maybe aging really is a circle, and that it's malleable, and we can not just accelerate it, which we were doing at the time. It's all published now. But we could even reverse that process. We could reset the epigenome if that's truly involved. And you can see here, I was writing things like resettable epigenome, and we were looking to use the Yamanaka factors potentially to do that. And of course, Belmonte, Juan Carlos Belmonte, deserves huge credit for being the first to do this in vivo, to show in vivo you could actually very carefully treat a mouse with Yamanaka factors and not kill it, and actually make it healthier and longer lived. And we have stood on his shoulders as well. I'm fascinated with cell identity. Of course, the epigenome gives cells their identity. It's what allows development to happen. It's how you get an embryo from a single cell. And the idea at the time was maybe we get what I came up with at the time was called epigenetic drift, that due to insults to perhaps the chromosome, DNA breaks, circles of DNA, these distract the epigenetic regulators, and what were once very powerful boundaries and accurate gene expression levels become smooth, and actually you don't get the beautiful pattern of gene expression. And I did some mathematical modeling of that helped my thinking. And then, inspired by Claude Shannon's work, I started going nuts about analog versus digital information and noise, and the observer, which you'll see later, and started to model biological noise in the same way that Shannon modeled informational and digital noise. And I started to realize there were two types of information in the body. There was digital information, which is DNA, and a lot of the epigenome, which is just as important for life and longevity, is analog. Analog information is stored in the body as structures of how the DNA is actually wrapped. And it's in 4 dimensions. It's not in one or two. And I started to think about correction data. Was there correction data really stored in every cell? And we started to look for it. And we were wondering, how do we go back, like Claude Shannon suggested, to not just slow down the noise, but actually reset the information in the cell. Here I'm saying maybe it's stored in the form of RNA and DNA. And the output of all of this system is life and longevity. And then I started to think about what makes us different and similar as species. And this was an idea where the epigenetic landscape, which gives beautiful gene expression, this is, of course, modeled after Waddington's landscape, is that current interventions are trying to slow down this bouncing of this virtual cell that's not trying, but can jump across valleys. But it doesn't when we're young, because these boundaries, epigenetic boundaries, which Manolis has beautifully defined, they erode over time, we think partly due to cell stress and broken chromosomes. Now we've got more entropy, less boundaries in the epigenome, and now you can see that cells can jump from one cell type to another. They don't really jump, but we do see them drift. And actually, experiments that we've now published show that older cells are easier to reprogram into different cell types because the boundaries of their cell type are lower. And perhaps I was thinking at the time, perhaps a species that lives a long time, like a bowhead whale or a Greenland shark, would have less vibration, fewer DNA breaks, but also greater stability of the boundaries that keep cells and maintain their identity, so that even an chronologically old Greenland shark, compared to us at the same age, at let's say 90 years old, would have greater ability. Well, perhaps here is what we would compare to this, has greater boundaries. And so I was looking for ways in my lab to slow down this process by increasing Sirtuin activity, increase DNA repair, protect the genome. We've now done that. Some of it's published. We have enzymes now and proteins that stabilize the worm genome and make worms live longer. We're doing this in mammals as well. But what about, can we truly slow down the erosion of these landscapes? And can we ever get this cell to go back to where it was? Is that information even possible? Now, making a stem cell using Yamanaka factors was not going to be the solution, because importantly, the difference between what Yamanaka did and what I'm trying to do here is that Yamanaka erased cell identity, basically took these hills and flattened them so that the cells had no identity. I'm trying here to do the opposite, regain the identity. So I started to call this process X-differentiation. Richard Cutler has called it dis-differentiation. And then the redifferentiation would be the reset. And this is a Waddington's landscape of the epigenome during development. And in this review and perspective with Wancheng Lu and Xiao Qian from my lab at the time, we've extended the Waddington landscape out into the future. So now it includes the adult and the old individual. And you could see that the cells that were once in a beautiful valley are now more likely to move around the landscape and lose their identity. And losing your identity is a real problem, right? You're probably more susceptible to diabetes, Alzheimer's, even cancer. And we've written about that as well. For cancer, it's called the gerontogenesis hypothesis. So this is more of a fully formed depiction of the theory that we've published. And of course, if anyone has read Claude Shannon's mathematical theory of aging papers in 1948, parts A and B, they will recognize this. I have stolen it and just replaced some of the boxes with biology rather than math and numbers. And it's similar, though, that our bodies are like information that gets transferred across time and space. There's noise that's introduced by damage to cells. And eventually the information is lost. And most of it is epigenetic and analog information, because analog information is much harder to maintain over a lifetime than digital. Of course, you'll appreciate that that's the reason we live in a digital world. The major reason is that it's less subject to noise. And even if you do lose digital information, you can go back with the TCP/IP protocol and find the missing bits with what Claude Shannon was calling the observer. Now we call it the backup copy. And we've been looking in my lab for, first of all, how do you contact the observer, and what is it made of? Where is epigenetic youthful information stored? And we have evidence that it exists, which is really exciting. So we use this paradigm to cure blindness in mice. We're now improving vision in blind monkeys at Life Biosciences here in Boston. And hopefully we'll, if all goes well, treat a human for blindness next year. Or if perhaps even multiple diseases, not just regular blindness, but perhaps glaucoma, macular degeneration, perhaps one day an entire human. But we're starting with the eye, if you're wondering, because for gene therapy, it's the best place to start. It's an enclosed, immune privileged location in the body.